All right, so before we get into the actual blood typing, I want to talk about just blood types in general. So we have 4 different blood types. We have A, which I'm going to write down here on this first red blood cell. We have blood type B, we have blood type AB, and we have blood type O. So what determines our blood types? And that is actually based on antigens that are found on the surface of the red blood cells, okay? So we have basically 3 different types of antigens. The first one we're going to call, which I'm going to symbolize in black, is antigen A. The second type of antigen that we have, which I'm going to symbolize in green, is antigen B. And lastly, our third type of antigen is an Rh antigen. And these antigens are found on the surface of our red blood cells, which is what we're going to look at here. So the surface of our red blood cell for blood type A has a bunch of A antigens on it. So I'm just going to put 4 of them here, just symbolizing that we have the A antigens on the surface of the red blood cell, making it blood type A. Blood type B has B antigens on the surface of the red blood cell. Blood type AB, okay, has both A and B antigens. So I'm going to put 2 A antigens on it and 2 B antigens on the surface of the red blood cell. So blood type O, well, we only have antigens A, B, and Rh. There are no antigens on the surface of the red blood cell in blood type O. When we talk about the Rh antigen, if it's present, means that it's a positive blood type. If it's absent, it's a negative blood type. So let's say that our AB blood type also had the Rh antigens, okay? That would make that blood type AB positive. Our blood type O has neither A or B antigens, and it doesn't have any Rh antigens, which would make our blood type O negative, okay? So those are the antigens that are found on the surface of the red blood cell. Within our blood, we also have antibodies. The antibodies are floating around the red blood cells themselves. We're not going to focus on those right now, just simply because that's not what we need for blood typing. So the idea here is that our blood type A has A antigens and it has B antibodies, which are floating. These are in the plasma. So anytime we see antibodies, they're going to be in the plasma, okay? Blood type B has B antigens, and it has A antibodies. Type AB blood has A antigens, it has B antigens, and it has no antibodies. Blood type O, once again, no antigens. And then the antibodies that it has both A and B antibodies. So whatever antigens it has the opposite antibody. So again, blood type A has A antigens on the surface and the B antibodies in the plasma. Okay, so again, that just kind of shows you what our blood types are, what antibodies are present. But again, for blood typing, that is not what we're going to be focusing on. Instead, we're going to be focusing on really is what is on the surface of the red blood cells, okay? So what we can do to determine our blood type, because if we have a blood sample, we don't know what's in it. What we need to determine is what antigens are on the surface of the red blood cell to determine what blood type it is. So we need to know, is there A antigens? Is there B antigens? Is there A and B antigens? Is there no antigens? And again, is there the Rh antigen? So basically we're looking for these 3, A antigen, B antigen, and the Rh antigen. So how in the world do we do that? Okay, so in this example, I'm going to give you guys and show you one well. Okay, so one of these wells, you'll notice that it has 3 wells, or one of these slides has 3 wells, an A well, a B well, and an Rh well. So in order to actually see the blood typing and how it's done, what I want to do is put it on a white background so we can see the 3 wells. Okay, so again, I'm just going to mark these on the piece of paper, A, Rh, and B. So what we're doing is we're testing for the antigens, for the A, the B, and Rh antigens in the blood itself. So the first thing that we're going to do is we're going to take our sample of blood. This happens to be sample number 1. There are 4 different samples that we're going to be looking at the results of, but this is the one that we're going to be testing. And I'm going to put 3 drops of just the blood in each one of the wells. So I'm not adding anything else to it at this point, okay? So what we're going to do and how we're going to determine which antigens are present is we are going to mix them with the antibody, okay? So the first antibody that we have in this blue bottle is anti-A. So this is antibody A. So we're going to mix it with the A well, okay? And what this antibody is doing is it is going to try to attack the antigen to see if it is present. So antibody A attacks antigen A and it causes agglutination or clumping. It can also look like a gel-like fluid. It can also cause a crystal structure to appear within the blood. So I added 3 drops of the blood and 3 drops of the A antigen in the A well. And the first thing I'm going to do is I'm going to mix those 2 with a plastic toothpick here, and I'm going to let that sit for a minute. The next thing that we're going to test is the B antigen, and we're going to mix it with anti-B serum. So this is basically a bunch of antibody Bs, okay? So again, if the B antigen is present, this anti-B antibody is going to bind to it and it's going to attack it. It is going to cause it to clump. It's going to cause it to agglutinate. So again, we're going to mix that sample. The last one that we're testing is the Rh antigen. So the green bottle here has Rh antibodies in it. Again, we're going to mix 3 drops of this with the 3 drops of the blood. And again, if the Rh antigen is present on the surface of the red blood cells, that Rh antibody that we just added is going to attack it and it's going to cause it to clump. So that's what we're going to be looking for. So when we're looking for all of this, the thing is, clumping can look like a few things, and I've mentioned that already. It can actually cause the blood to become clumpy. If we look at this one really carefully, you can actually see the little tiny specks in it. That is a positive agglutination reaction. If we look at well B, the color might be a little different, but it's still fluid. There's nothing in it. So that's actually a negative reaction. And when we look at the RH is a little hard to see, but I have in the RH well, I actually have these little tiny clear crystal-like specks in it. So that's telling me that I have a positive result in the A well, a negative result in the B well, and a positive result in the Rh well. So what in the world does that mean? It means that antigen A is present, so we know that it has antigen A in it. Antigen B is absent. So if antigen A is present and antigen B is absent, so far we know that we have a blood type of A. The Rh well showed a positive agglutination, which indicates that we have a positive blood type. So again, we have a positive in the A well and a positive in the Rh. That gives us a blood type of A positive. Let's give another example, okay? What if we had agglutination in well A and in well B? That would mean that antigen A and B are present, in which case it would be blood type AB. What would happen if the Rh clumping or agglutination was negative? That means that it would be an AB negative blood type, okay? So the idea here is wherever you have agglutination, that's the antigen that's present. So if it's present, it means it's there. So if you have a positive result with antigen A, it means that, or in the A well, it means that antigen A is there. If you have a positive result in the B well, that means that antigen B is present. If you have the positive result in Rh, it means that the Rh antigen is there. If you have a negative result, it means it's not there. So again, with this specific blood type that we're looking at, we had a positive result in A, which means we have blood type A. Negative result in B, which means B wasn't there, so it's just blood type A. And again, we had a positive result in the Rh well, which gives us our positive blood type.